Hello students, welcome to Turito live classes. Myself A. Swagat. Today in this particular session, we will be having an understanding about digestion process. So, we will understand the whole of the process, how our food gets into the stomach and it proceeds further for the activity of digestion. So, that is what we will be proceeding with further. So, process of digestion, that is the topic for today. So, if we look here, we would like to have a topic what we have dealt in the previous session. So, that is what we will recollect and then we will be looking into the objectives. So, in the previous session we were having a detailed understanding of structure, location, secretion and functioning of various digestive glands. So, that is what we have been doing in the previous session, starting with salivary glands, liver and pancreas, apart from which I also explained you about some of the secretions released within the stomach. The stomach, even though it is not a gland, even then it releases some secretions and that help us to proceed further with the process of digestion. Here in this particular class, we are going to learn about digestion as an activity. It converts the complex food components what we consume into a simpler ones. So, that is what is all about digestion is, and then we will also look into the various parts involved in the digestion process. So, before having the detailed understanding of the digestion process, we do have to look at the digestive system. So, when we look at the digestive system, that will enable us to understand what we have dealt earlier and thereby making us to recall the topics. So, we will also look into the process of peristalsis, which helps the propelling of bolus to the stomach, followed by chemical and mechanical digestion which is taking place in the stomach. So, we will recall the digestive glands and their respective secretions, followed by completion of digestion process in the intestine. So, specifically the digestion process gets completed in the small intestine, right. So, can you tell me which part of the small intestine we see the digestion gets completed? Which part of the digestive system, that is which part of the small intestine the digestion gets completed? Okay, some of you are saying jejunum. I will write your responses here. Okay, ileum, and you said what is that? Yeah, I got it. Duodenum. So, actually this is the first part of the small intestine, that is where you will see the actual digestion gets completed, okay, and then we will see that the digested components in the small intestine would be completely absorbed. So, if you see here, digestion is a process which break down the large complex insoluble organic food components, see insoluble food molecules, because we consume different types of foods, right. So, you certainly cannot expect that complex food made, let it be a plant component or it is a animal component or whatever it may be, you cannot expect it to just get digested just by putting in the mouth. So, you cannot see that it just gets digested when you just by dropping it in water or in any of the solution. It is not so easy. So, it needs a very complex system to make sure the big chunk of food what you put in your mouth, okay, that gets completely digested and forms ultimately into nutrients, which is very very small compared to the chunk of food what we put in our mouth. So, if you observe, so simpler soluble and diffusible particles by the action of digestive enzymes. So, that complex insoluble inorganic food organic food needs to be changed into simpler soluble and diffusible particles. That means you can understand at the cellular level how diffusion occurs. Now, at cellular level the diffusion intensity and the concentration needed, everything is you are aware of it. So, to that level the food which you put in your mouth has to be changed, that has to be converted. So, that process which happens here is digestion, that also occurs with the aid of the enzymes present in the body. So, that is what was all about digestion. So, it is like breaking of the complex, very complex food components, what you eat, the food items what you eat, okay, they have to be converted into the ultimately the food components, which is nothing but the carbohydrates, proteins, fats. So, they are known as food components. Carbohydrates, proteins, fats, vitamins and minerals. So, these are the food components, whatever we consume. From that we have to make sure our system ensures that all these components are separated properly, and all these components ultimately they have to get absorbed. So, they have to get absorbed. Now, that is what happens. Now, if you have to understand this how happens, so we have to look into the system how it looks like. So, it apparently looks like this. So, this particular image is not new thing to you, right. So, we also had a look at it in the previous session also. Now, if you observe, so this is actually that bag which takes all of our bolus into it, okay, and there are other organs like liver, you have gallbladder, duodenum, that is the first part of the small intestine, followed by jejunum, and then you would see end up having the longest part of all is ileum. So, our small intestine is starting with duodenum, and it has second part known as jejunum, followed by ileum. So, it is in the ileum actually, starting from jejunum to ileum, whatever food components that have been digested ultimately reached their the basic units, their respective basic units. That is where you will see the ileum is taking care of them in order to get them absorbed. So, that process done there, right. So, of course, we will be looking into the functioning of the pancreas, gallbladder and the liver. Okay, I will show you what happens out there. Of course, we have to begin our discussion with the stomach, right. So, we will do all those things in this particular session. So, I just wanted you to have a look at this whole system and just recollect what we have dealt earlier in using this particular image. Okay. So, you can understand that here what you are looking is quite simple, the diagram in the image. So, make sure you guys all have the practice of drawing it. So, regularly you have to practice it, and if you try to put your answer with a diagram, okay, that is more expressive than your words. So, that is always important. So, that is one good thing what I want you to tell. So, fine, let us move forward. So, I would like to show you starting with this, the salivary glands. So, can you tell me how many salivary glands we have? Okay, that is correct. So, you see that excuse me children. Okay, so you say the count is 3, okay, and you are also saying that we have different types. So, parotid gland, okay, and we have excuse me children. So, these are the 3 types of glands present in the buccal cavity. So, when you look at a buccal cavity, These are all are the types of the different types of salivary glands what we have. All of them have the same function, that is to release the that saliva which contains ptyalin. It has lysozyme, okay. It has salivary amylase. So, all of those excuse me children. Yeah. So, all of those which are released here, and we see that they perform this particular function of digesting the complex carbohydrates. You see the parotid gland here. Next is the sublingual gland, submaxillary or submandibular gland. So, these are the three types of gland present in the buccal cavity, arranged in an order. Excuse me children. Excuse me. Yeah. So, these three types of glands which are present here, they have specifically tube like arrangements from them, so that using those tube like structures, that is the ducts, they release the salivary amylase. So, that is one thing we have. So, this is what we have looked into the previous session, we looked at it, and we also used very same buccal cavity having the tongue which helps us to taste the stuff. So, we saw that tongue is helping us to taste different types of tastes, having sweet, salty, sour, bitter and umami. So, the essence of the taste itself can be sensed much before consuming the food. So, that essence of taste, that is also called as umami, that itself is a considered as another taste. Now, coming to the buccal cavity, and we just looked into the different types of glands, salivary glands. So, our concern is something about this. It is about those components when we consume and we sense the sweetness. It is for those components that would start their digestion right in the mouth. That is with the action of the teeth when we chew the food, okay, along with the muscular tongue we have. It also helps in properly orienting the food along with the location of the different types of dentitions what we have, in the sense like different types of teeth what we have. Based on that, our tongue reorients the food accordingly and we chew it, and ultimately the food which gets combined with the saliva, and you get to see that terms itself to form bolus. So, this is a simple process, it forms the bolus. Now, it is the bolus that has to pass through the very small tube which is present, regulated by some stuff I would like to show you. So, that is what is esophagus. So, that is where you see. So, here if you observe, these are the three glands, okay, we have based on the arrangement, followed by a viscous fluid with a pH of 6.7, and this is what we have discussed in the previous session. Around 1 to 1.5 liters of this fluid is released every day, and we see it consists of water, salts, mucin and ptyalin. So, this amylase and lysozyme are the main enzymes which acting there in order to get themselves work together for digesting the carbohydrates first of all, and then lysozyme is one good enzyme present in the saliva, helping in breaking apart the bacteria or any other bacteria like stuff entering into the food. When we consume that, it just happens when we try to enter that food, and it often happens. So, that is the reason why we have lysozyme and amylase working here, so that it just directly involves in the process of that. So, when we have this complex starch, So, it is already mentioned here digestive carbohydrate. So, the starch is actually a complex carbohydrate. So, this complex carbohydrate starch by the action of salivary amylase gets converted into maltose. So, this maltose is also quite complex than glucose, but it is maltose is simpler than starch. So, maltose is simpler than starch. So, starch is very complex carbohydrate. It gets converted into maltose by the action of the salivary amylase, and our maltose has 2 units of glucose. So, when you combine 2 units of glucose, one glucose another glucose, when you try to combine it, you get to see that it is actually forming the salivary glands. It is actually forming the simpler carbohydrates. Now, this is what happens in the salivary glands, following which we see that peristalsis, like I mentioned earlier. So, peristalsis is also one of the important thing to be understood. So, in this peristalsis, what we have mentioned and we have already learned previously. So, from the esophagus, the bolus further passes down by the successive waves of muscular contractions. So, that muscular contractions, children, is what is peristalsis, and we know it looks like this. That movement is done by the peristalsis, and we see there are regulators present here. You cannot expect that the food directly entering into the esophagus just like that, it is not possible. So, there lies a regulator here, that is the sphincter muscle, at the top of the esophagus, just at the beginning of the esophagus. The opening, the beginning, the moment when you see that the bolus is trying to enter the esophagus, there happens to be a small opening, right. That opening is known as gullet. That gullet is known as that gullet is where you will see a sarcopharyngeal sphincter first of all. Okay. So, that is where you will see sarcopharyngeal sphincter, that is the one which regulates the movement of bolus into the esophagus, following which when the bolus is about to enter the stomach, we will see gastroesophageal sphincter regulating the movement of the bolus into the stomach. So, you cannot expect this peristalsis, it does not move the complete bolus all at once to the stomach, it is not like that. It just passes the bolus bit by bit into the stomach, because the digestion which has to occur, it has to happen in a process, and it has to get most of the stuff gets digested, right. So, that is why we have to chew the food properly, so that you would end up having most of the carbohydrates digested in the buccal cavity itself. Later, the second step, when the food is entering our stomach, that is where you see the proteins are getting digested. So, if you have to allow that to happen, this is the system in which you see sarcopharyngeal and gastroesophageal sphincter regulating the movement of the bolus, and you would see this is how the contraction and the regular relaxation makes the bolus to move into the stomach. That is a continuous process. Now, in the stomach you will see there happens the digestion of the food, that is what you see, that is digestion by stomach. It is the bolus that has to enter the stomach, and the bolus has to get digested. Now, it is not so simple to get the bolus digested. So, here the digestion of stomach happens in 2 ways, one is a chemical digestion, another is a mechanical digestion. Okay. So, when the chemical digestion happens, it also coupled up with the mechanical digestion, even though for our convenience we are looking here that chemical digestion, mechanical digestion given separately, but actually there are chemicals released inside the stomach, they act on the bolus, and the mechanical digestion, in the sense the bag like structure what you have seen, it has to be moving continuously. That is why you would see that the bolus is get itself mixed up with all the gastric juices released in the stomach, especially hydrochloric acid and all other stuff. So, you see that we have mucus and pepsinogen in helping taking care of the chemical digestion, followed by churning of the stomach. That is, the muscles of the stomach which are present there, they also would be moving continuously. So, their continuous movement of the continuous process throughout the process, that helps them to get the food digested. Now, here I have to tell you, we have seen different layers of the stomach, right. Now, here the stomach releases one of the most important enzyme here, that is pepsinogen. And this is not an activated enzyme. This pepsinogen is an inactivated enzyme. So, it is an inactivated enzyme. Now, when it is an inactivated enzyme, it has to get activated, and that is when you see it starts acting on the bolus. You agree with that? Okay, correct. So, can anyone tell me how does it gets activated? Any guesses? Okay, correct. Some of you are saying. So, can you tell me, can anyone tell me what is the function of mucus here? Mucus is also given out. Okay, correct. So, basically mucus helps in protecting stomach, protecting lining of the stomach, isn't it? So, from whom it protects? What is that thing? Why mucus has to protect the lining of the stomach, internal lining of the stomach? Correct. So, as you have dropped in correctly, it is from the action of the hydrochloric acid, mucus will be protecting the stomach. But where does this hydrochloric acid came from? Where did we get this from? Of course. So, like you have dropped in, it is from the stomach, right. So, when you see it is from the stomach, you see when hydrochloric acid is getting combined with pepsinogen, it is then you will see that the inactivated form, that is pepsinogen, gets activated, then it begins the action of the digestion, right. So, pepsinogen is an inactivated form of pepsinogen, sorry, pepsin. It is an inactivated form of pepsin, okay, and that pepsin is an enzyme that acts on the food, that is the bolus, in order to convert the proteins, the complex proteins, into simpler units of the proteins. So, here you see the bolus of the food remains in the stomach for about, this is the timing, and it gradually converted into a semi fluid mixture. So, this is nothing new to you, we have already learned it, right, semi fluid mixture, that is chyme. So, that is fine, but what else we have? This pepsinogen, it is a proenzyme, in the sense it is an inactivated form of an enzyme, okay. When it gets exposure to the acid, that is hydrochloric acid, that is when you see it is turning itself into pepsin. Now, this pepsin, when it acts on the bolus, it will try to convert all the proteins in the bolus into proteases and peptones. So, that is what is happening in the stomach. So, in order to convert this pepsin, or in order to make the pepsinogen activated to pepsin, we need pepsinogen to get exposed to the hydrochloric acid, and then it turns itself into pepsin, and that pepsin acts on the proteins present in the bolus. So, this combination of the bolus and the gastric juices along with mucus and hydrochloric acid, whatever you have seen, that turns out to form a liquid mixture, that is chyme. So, that is when we call it as chyme. So, you see that the mucus and bicarbonate plays an important role in taking care of the mucosal layer of the stomach, epithelial layer is the outermost layer of the stomach. So, you see that. So, that is why we need that. So, this is what happens there. This is something exception here children, I wanted to learn here, that is why I mentioned it here. When you see infants, what is their food, the primary food what they consume, infants, baby less than 2 years or 1 years, obviously, right. So, they like to consume milk, right. Now, milk has specific type of protein, okay. That protein is to be digested by a specific enzyme called renin, R-E-N-I-N, okay. Now, this is specifically found in only infants, which helps in digestion of milk proteins. Not so easy to digest those milk proteins. That is why this renin is present in the case of infants, so that they can digest the milk proteins. Now, you might be wondering, sir, we do consume milk, many of us consume milk, but how we are able to digest those proteins? That might be a question, right. The milk what we are consuming, we often consume soon after boiling, right. So, that heat is more than enough to denature the proteins which are present in the milk already, okay. So, there is an action of temperature happening on those proteins before we consume. Even if you take the milk which you get in the market, they are packed. So, I am sure you all are aware of the process of pasteurization. So, that is there also you see that the milk gets processed, right. So, in which the proteins are altered. So, when we are consuming that, our body is more than enough capable to digest those milk proteins, but in the case of infants you cannot expect we to give them boiled milk. So, it is not possible. So, directly get the milk from their respective mothers. So, it is for that reason they must have this renin in order to digest that readily available milk from the mother. So, lipase is also secreted in small amounts, and that is also included in the digestion process. So, here you see here proenzyme pepsinogen on exposure to hydrochloric acid converts itself into pepsin. You see pepsinogen right converts into pepsin when it is exposed to hydrochloric acid. So, pepsin is active form of the enzyme, and then you see when these proteins from the bolus gets exposed to pepsin at this particular pH, because HCl is present, that turns out to be proteases and peptones are formed, right. So, they have the smaller units of proteins are peptides. Peptides are the smaller units of the proteins, right. So, that is the kind of arrangement what we have right now. So, you see digestive glands, right. So, hope you understood how the digestion occurs in the stomach, right. Now, what you are going to look at is the digestive glands. So, this particular image what you are looking at is definitely not new to you anyway, right. So, here you observe the structure, that is liver, we have gallbladder, okay, we have the duodenum, we have the pancreas, okay. So, this is the main focus where we are going to have a look at. So, hope you can tell me the different components secreted or produced by these glands. So, what do we get from this liver? Who will tell me? Drop in the chat box quickly, come on. Correct. So, liver actually secretes the bile juice, okay, and you see that bile juice. What about gallbladder? What is it doing here? What is it doing here? Gallbladder. Correct. So, here so bile which is released, which is actually produced by the liver, would be stored in the gallbladder because the liver is one of the fantastic organs. It is the only organ here what we can find that keeps its ability to regrow. Sorry, it is the only gland actually, I mean it is one such gland keeps the ability to regrow. Now, when it is able to regrow, it is a very big gland, the largest gland of the body is liver, it has so many other functions to perform. One such function, what is done by it is producing bile. So, whenever the food enters the duodenum, okay, this part so duodenum is the first part of the small intestine, and that is where you will see that the food gets digested. Okay. Now, it is the case, what you have to understand is, when the duodenum needs the bile juice, that is a point when you cannot expect liver to directly produce the bile. It is for that reason it is given with a gallbladder, where it stores the bile juice, and when there is a need of the bile juice here at the duodenum, it just enters into this particular part using a common bile duct, and that is how it happens. And when it comes to pancreas, they also release certain enzymes. So that you might have heard it earlier, they release some of the substances and we call them as complete gastric juice or complete digestive enzyme, because the secretions given by the pancreas are designed in such a way that they are composed of different kinds of enzymes, which keeps the ability to digest primarily carbohydrates, proteins and fats. When they have to do that, definitely their secretions must have all types of digestive enzymes. Let us move forward. So, digestion in the intestine, that is what we have to understand. So, trypsinogen is one such thing which is released by the pancreas. So, trypsinogen is released by the pancreas. Okay. Now, it has to get activated, right. If it has to get activated, it needs an enzyme called enterokinase enzyme. So, this enzyme acts as a catalyst, okay, in order to convert the trypsinogen into trypsin, which is again an active form. So, trypsinogen is an inactive form. So, trypsin activates other pancreatic enzymes. So, that is one good thing why trypsin is given here. So, it is a reason when it can sense, pancreas can sense the presence of the chunk of chymes entering the duodenum, and duodenum is the first part of the small intestine where the complete digestion of the food happens. Now, that is to be sensed by this, and this process happens. Now, in the pancreas there are different kinds of enzymes released. So, they are you will see amylases, proteases and lipases. So, are present in the pancreatic juice. So, pancreatic juice have amylases, proteases and lipases. That is why you can understand by looking at it, this can digest carbohydrates. So, I will be writing here anyway for you. Okay. So, amylases can digest carbohydrates, proteases digests proteins, okay. Lipases keeps the ability to digest fats. Okay. So, that is the picture here of the pancreatic juice, and this pancreatic juice is actually the pancreatic juice actually is the complete digestive juice. Okay. Now, that is how it happens. Following which, yeah. So, it is because of the presence of these enzymes, okay, the complete digestion of the proteins finishes by the time when the chyme reaches the first part of the small intestine, that is duodenum. Okay. Now, remember children, so our digestion of carbohydrates is completed again because of the amylases are present in the buccal cavity, and also they are present in the pancreas. Our problem of the proteins is also solved because proteins were also gets digested in the duodenum. okay, that is fine. And much before that you see proteins were started digesting in the stomach, in the acidic medium, fine. Now, the moment when you see that they are entering into the duodenum, they are surely in the acidic medium, and you see the pancreatic juice is also released. Okay. Now, when that is released, you see that proteins are also completely digested, carbohydrates are digested. So, both are done. Now, our main problem remains here is with the fats. How we are able to digest the fats is a thing. So, you may think, sir, we have lipases, so lipases would help to convert the fats or to digest the fats. Yes, lipases are designed to convert the fats into simpler ones, fine, but lipases all alone are insufficient. So, this lipase is insufficient. Okay. So, when it is insufficient, we have to find out an alternative. So, what is the alternative? What you can make out here? Hope you understood the picture now. Correct. So, as you have already thought of, the picture comes up with this. So, that is actually the bile juice which actually comes into the picture here, that bile juice combines with the lipases. Okay. So, that is when it actually converts the acidic medium to basic medium. Okay. That basic medium in the duodenum, and you see that this is where in the duodenum emulsification of fat happens. So, emulsification of fat occurs in the duodenum with the help of bile juice, okay, along with lipases. So, that has to happen in a different medium than the stomach, and that medium is basic, also known as alkaline medium, right. So, hope you understood this. So, proteins, proteases, peptones in chyme reaching the intestine are converted into dipeptides, right. So, that is what exactly happens in the small intestine. So, proteins are digested, and you know emulsification of fats was done, and ultimately that happened in the basic medium, and that is when you saw that the complete digestion was done. So, when the digestion was completed, you see you have already seen the structure of the finger like projections in the small intestine, that is microvilli, and that is where you will see the absorption of the nutrients or absorption of the digested food happens. Okay. And the food which is not getting digested, okay, or the undigested food, okay, you see that undigested food that is to be stored in the rectum. So, that food will be stored in the rectum after passing through the large intestine, where in the large intestine water reabsorption happens. Right. So, water reabsorption happens. Now, that is when it happens, and we will see that after the water reabsorption, the undigested food gets stored in the rectum, and that is what we know it as fecal matter. Okay. And that fecal has to get ejected out of the body, right. That undigested food which is stored in the rectum and that gets ejected out of the body through anus. This process is known as ejection. This is what happens when the digestion process completes, and you see that the undigested food is ejected out of the body. So, hope you understood the session as a whole, and we will move forward here to look into the summary. So, we looked at the digestion activity as a process to convert the complex food components into simpler ones, and we recall various parts involved in the digestion process. We used the digestive system image to recall that, following which we looked at the propelling of the bolus into the stomach through a process of peristalsis. And then we looked at the chemical and mechanical digestion in the stomach, following which we recalled the digestive glands and their respective secretions, which help actually in the process of digestion. Following which we looked at the complete digestion process and how it gets absorbed in the small intestine and the undigested food gets ejected out of the body, right. So, this is what we have observed in this particular session. So, hope you understood this session. Let us meet in the upcoming session and we will discuss a lot many topics further. Thank you everyone.